Hello there. Tutor, it's great to connect for another deep dive session. Today, I'm really keen to thoroughly explore a topic that's, well, not just fundamental to biology, but absolutely essential for our class 11th and 12th board exams and critically important for competitive exams like NEET. We're going to unravel the complexities of photosynthesis in higher plants.
Namaste. Yes, you've picked the winner. Photosynthesis is more than just a chapter. It's, the very foundation of life on Earth. Think about it. Every bite of food you eat, every single breath of oxygen you take owes its existence to this incredible process. So our mission today is to demystify it, transforming these complex scientific concepts into clear, exam-ready knowledge. We'll meticulously break down its core mechanisms, trace the fascinating historical discoveries, dissect the intricate machinery involved. We'll delve deep into its two main phases, the light and carbon reactions, understand the distinct pathways like C3 and C4, and finally examine the environmental factors that regulate its pace. Mastering this chapter will definitely give you a significant edge in your upcoming exams, so let's jump right in.
Foundations of photosynthesis, the basics, and early discoveries.
Okay, let's start with the basics. What is photosynthesis? At its core, it's a physicochemical process. What does that mean? Well, it involves both physical elements like light energy absorption and a series of sophisticated chemical reactions. Essentially, green plants use light energy to synthesize organic compounds, basically, their own food. This ability to produce their own sustenance earns them the label autotrophs. Makes sense, right? Auto means self. Troph means nourishment. And we, along with all other organisms that rely directly or indirectly on these plants for nutrition, are called heterotrophs. Now, why is this process so monumentally important? There are really two primary reasons. Firstly, photosynthesis is the ultimate source of food for virtually every living form on Earth. Whether you're directly consuming a plant or eating an animal that fed on plants, that energy ultimately traces back to the sun, captured by photosynthesis. It's the base of the food chain. Secondly, and equally crucial, it is uniquely responsible for releasing molecular oxygen into our atmosphere. This oxygen, as you know, is indispensable for respiration, the process by which most living organisms extract energy from food. Just pause for a moment and truly consider the implications. What if there were no oxygen to breathe? It profoundly underlines the sheer importance of photosynthesis, doesn't it?
That makes perfect sense. I do remember some basic experiments from earlier classes, but how did scientists first figure out what plants actually needed for photosynthesis to happen? What were those initial clues?
That's a sharp question, and it takes us back to some remarkably simple, yet really elegant experiments that formed our earliest understanding. You might even have performed variations of these yourself. They clearly demonstrated the basic requirements for this process. Think about the classic variegated leaf experiment. A leaf that has both green and non-green patches. If you take a leaf like that, or even a normal leaf partially covered with black paper, expose it to light, and then later test it for starch. Well, starch indicates photosynthesis occurred. You'll find starch forms only in the green light-exposed regions. This single experiment cleverly proved two things simultaneously. That chlorophyll, the green pigment, is absolutely necessary, and that light is indispensable for the plant to make its food. Then there's the experiment to show the need for carbon dioxide. Picture this. You isolate a part of a leaf by enclosing it in a test tube containing cotton soaked in potassium hydroxide, or KOH. Remember, KOH is a strong absorbent of CO2. It just sucks it out of the air. The rest of the leaf remains exposed to the open air. After placing the setup in light for some time, if you test both parts of the leaf for starch, the exposed part will test positive, but the part inside the tube with KOH will test negative. This unequivocally proves that carbon dioxide, CO2, is a non-negotiable requirement for photosynthesis. Simple, yet powerful observations. Now, our current sophisticated understanding of photosynthesis didn't just, appear out of nowhere. It's the result of centuries of scientific inquiry, with scientists building upon each other's discoveries. Let's trace this fascinating historical journey, as it gives crucial context. It's actually quite interesting. We begin with Joseph Priestley back in 1770. This remarkable scientist, who, by the way, later discovered oxygen, conducted experiments with a burning candle and a mouse in a closed bell jar. He observed that both the candle would soon go out and the mouse would suffocate. He concluded that burning and breathing somehow damaged the air. Something was being removed or something bad was being added. But here's where his genius shone. He placed a small mint plant inside the same bell jar with the candle or mouse. And to his surprise, the candle continued to burn and the mouse stayed alive. Priestley hypothesized that plants restore to the air whatever animals and burning candles remove. A foundational insight, really. Building on Priestley's work, Ingenhousz entered the scene in the late 1700s. He used an aquatic plant, something like hydrilla, and observed that in bright sunlight, tiny bubbles formed around the green parts of the plant. But these bubbles vanished in the dark. He deduced that sunlight is crucial for plants to purify the air, and critically, that only the green parts are capable of releasing oxygen. That was a key step. Fast forward to 1854, and Julius von Sachs provided concrete evidence that glucose is produced when plants grow, and that this glucose is often stored as starch. More importantly, he pinpointed the location of the green substance chlorophyll within specialized bodies inside plant cells. We now know these as chloroplasts. He effectively identified them as the sites of glucose production, locating the chemical factory, so to speak. Then we have the truly ingenious experiments of T.W. Engelmann in the late 1800s. He used a prism to split white light into its spectrum, like a rainbow. He then shined this split light onto a green alga, cladophora, which was immersed in a solution containing aerobic bacteria. Now, these bacteria need oxygen, right? So they would naturally gather where oxygen was plentiful. Engelmann observed that the bacteria overwhelmingly accumulated in the blue and red regions of the light spectrum. Why is this significant? This was the first action spectrum of photosynthesis. It directly showed which wavelengths of light are most effective. And remarkably, it showed that the most productive light for photosynthesis correlated with the light absorbed by chlorophyll A and B. Finally, a truly groundbreaking contribution came from Cornelius van Niel, a microbiologist, studying purple and green sulfur bacteria in the early 20th century. He showed that photosynthesis is fundamentally a light-dependent reaction where hydrogen from a suitable compound reduces CO2 to carbohydrates. His general equation was something like 2H2A plus CO2 yields 2A plus C0 plus H2O using light. His crucial inference was this. In green plants, water, H2O, is the hydrogen donor of the H2A. When it's oxidized, it releases O2. But in some bacteria, like sulfur bacteria using H2S as the donor, the product is sulfur, not oxygen. This led him to the profound conclusion that the oxygen released by green plants comes from water, H2O, not from carbon dioxide, CO2. This hypothesis, initially revolutionary, was later definitively confirmed using radioisotopic tracing, using heavy oxygen isotopes like O18. Powerful stuff. So, based on all these crucial discoveries, we now have the universally accepted overall equation for photosynthesis in higher plants. 6CO2 plus 12H2O yields C6H plus O6 plus 6H2O plus 6O2. And this happens, of course, in the presence of light energy. This equation elegantly summarizes the inputs and outputs. You can clearly see the carbon dioxide and water going in, and then the glucose, the food, some water, and that vital oxygen coming out. And remember that crucial detail from Van Niel's work. The O2 we breathe, the byproduct, comes entirely from the water molecules, not from the CO2. It's a key concept, often tested. And it's important to remember this single equation is just a summary. It represents a complex, multi-step process, not just one single reaction.
Wow, that historical journey really paints a vivid picture of scientific progress. It wasn't just one person, but building blocks over time. But if it's such a complex, multi-step process, where exactly does all of this intricate chemistry take place inside the plant? Can you help me visualize the location a bit better?
Absolutely. You're asking a fundamental question, because understanding the where is vital to grasping the how. While we commonly say photosynthesis occurs in the green leaf, it's more accurate to say it occurs in the green leaves and, well, other green parts of plants too, like young stems sometimes. Within the leaves, the specialized mesophyll cells are literally packed with numerous chloroplasts. You find lots of them there. These chloroplasts are the actual powerhouses, the tiny factories of photosynthesis. And here's an interesting detail about their efficiency. Chloroplasts aren't just scattered randomly inside the cell. Oh no. They strategically align themselves along the walls of the mesophyll cells to capture the optimum amount of incident light. That's quite clever. For example, under very high light intensity, they might arrange their flat surfaces parallel to the cell walls, sort of hiding to minimize photodamage. Conversely, in low light, they might align perpendicular to the incident light to maximize absorption. It's a dynamic, adaptive system. Now let's zoom in on the chloroplast itself. You might recall its structure from your cell biology chapter, right? Inside the chloroplast, there's a sophisticated membranous system. This includes stacks of disc-like structures called grana. Each stack is a granum, and the individual discs are thylakoids. And then there are stroma lamellae, which are flat membranous tubules connecting different grana. All of this membrane system is embedded within a fluid matrix called the stroma. What's truly fascinating and crucial for your exams is the clear division of labor within the chloroplast. The membranous system, so the thylakoids, the grana, the stroma lamellae, is entirely dedicated to trapping light energy. This is where the initial energy conversion happens, leading to the synthesis of those high-energy chemical intermediates, ATP, adenosine triphosphate, and NADPH. These reactions are directly driven by light, so we call them the light reactions, or sometimes the photochemical reactions. The stroma, on the other hand, that fluid-filled space, is where the enzymatic reactions occur. These enzymes take the ATP and NADPH produced by the light reactions and use them to synthesize sugar, which can then be converted into starch for storage. These reactions are, by convention, termed dark reactions or carbon reactions. However, and this is a common trick question in NEET, calling them dark reactions is a bit of a misnomer. Listen carefully here. They are not directly light-driven, that's true, but they are absolutely dependent on the products of the light reactions, namely ATP and NADPH. So they can and do occur in the light as long as the light reactions are supplying these energy carriers. If light disappears, the dark reactions continue only until the preformed ATP and NADPH are used up. Then they stop. This distinction is vital for a nuanced understanding. They need the products of light, not light itself directly.
The light-dependent reactions harnessing solar energy.
Okay, so we know where photosynthesis happens. Now let's get into the specifics of how plants actually capture that light energy. Have you ever wondered why leaves exhibit so many distinct shades of green, even on the same plant, rather than just one uniform green?
Yes, I have. It's like a whole spectrum of greens, isn't it? I always kind of assumed it was just one main green pigment responsible for everything.
That's a very common thought, but it's precisely where our deep dive reveals something quite intricate. The color we perceive in leaves isn't from a single pigment, but actually a fascinating blend of several. We can separate these pigments using a lab technique called paper chromatography. You might do this in a practical session. And it typically reveals 4 main photosynthetic pigments in higher plants, each contributing to that rich palette of green. First, we have chlorophyll A. This pigment appears bright or blue-green on a chromatogram. It is the chief pigment, the main player directly involved in the light reactions. Then there's chlorophyll B, which appears yellow-green. Next, we find the xanthophylls, typically yellow, and finally the carotenoids, which range in color from yellow to a vivid yellow-orange. Think carrots, that sort of color. So what exactly are pigments? Simply put, they are substances capable of absorbing light at specific wavelengths. Each pigment has its own unique absorption spectrum, like a fingerprint, showing which colors of light it absorbs most efficiently. If you examine the absorption spectrum of chlorophyll A, you'll notice it strongly absorbs light in the blue and red regions of the visible spectrum. It doesn't absorb green light very well, which is why leaves look green. The green light is reflected or transmitted. Now, what's truly significant is how this relates to the action spectrum of photosynthesis. The action spectrum measures the actual rate of photosynthesis at different wavelengths. And guess what? The action spectrum also peaks predominantly in the blue and red regions, showing a strong correlation with chlorophyll A's absorption. This clearly indicates that chlorophyll A is indeed the primary pigment driving the process. But what about chlorophyll B, xanthophylls, and carotenoids? These are called accessory pigments, and they play crucial, multifaceted supporting roles. They're incredibly important for several reasons. Firstly, they absorb light at wavelengths different from chlorophyll A. This effectively broadens the spectrum of light that the plant can utilize for photosynthesis. So light that chlorophyll might miss can still be captured. Secondly, they act like an antenna system, transferring the absorbed energy to chlorophyll A. Think of them as light collectors, gathering energy from a wider range and funneling it efficiently to the main reaction center, chlorophyll A. And thirdly, which is critically important for plant survival, they protect chlorophyll A from photooxidation. In excessively high light, chlorophyll can get damaged. These accessory pigments help dissipate that excess energy safely, safeguarding the primary pigment. Very important.
That's truly amazing how all these different pigments work together so precisely. It's like a well-coordinated team. But once this light energy is absorbed and funneled, how is it actually converted into useful chemical energy? What are the detailed steps of these light reactions you mentioned earlier?
Excellent question. And this is where we delve into the core of energy conversion, often called the photochemical phase. This phase involves several key sequential events: the initial absorption of light, the remarkable splitting of water molecules, the release of oxygen as a byproduct, and the formation of those high-energy chemical intermediates we touched upon, ATP and NADPH. Now, these pigments we just discussed aren't just floating around randomly. They're highly organized into efficient structures called light harvesting complexes, LHCs. These LHCs are integral parts of 2 distinct photosystems, photosystem I, PSI, and photosystem 2, PS2. A common point of confusion, so remember this. They are named based on their sequence of discovery, not the order in which they function during the light reactions. PS2 actually functions before PSI in the main electron flow. Each LHC is a sophisticated assembly of hundreds of pigment molecules chlorophyll A, chlorophyll B, carotenoids, all bound to specific proteins. They act like vast antennae, efficiently capturing light energy over a broad spectrum. This captured energy is then funneled, passed along from pigment to pigment, until it reaches a single specialized chlorophyll, a molecule. This molecule forms the reaction center of each photosystem. Each reaction center is unique in the wavelength it absorbs best. In photosystem I, PSI, the reaction center, chlorophyll, absorbs light maximally at 700 nanometers, and so it's called P700. In photosystem 2, PS2, its reaction center has a maximum absorption at 680 nanometers, and thus is known as P680. Okay, let's take this step by step to understand the flow of electrons, often referred to as the Z scheme. Why Z scheme? Because when you plot the electron carriers according to their redox potential, the pathway looks like a sideways letter Z. It helps visualize the energy changes. This electron transport chain is where light energy is truly transformed into chemical energy. Step 1, PS2 excitation. It all begins in photosystem 2. When the reaction center chlorophyll A, P680, absorbs a photon of 680 nanometer red light, its electrons become excited. They gain enough energy to jump to a higher energy level. This leaves P680 electron deficient or oxidized. Step 2, primary electron acceptance. These highly energetic, excited electrons don't just hang around. They're immediately picked up by a molecule called the primary electron acceptor. This happens very quickly. Step 3, electron transport system, ETS. From the primary acceptor, these electrons are then passed downhill. Now, downhill here refers to their energy level or redox potential. They move through a series of electron transport carriers, including cytochromes, progressively releasing energy. These electrons are essentially passed like a baton in a relay race. Step 4, BSI excitation. Meanwhile, and more or less simultaneously, in photosystem I, the reaction center P700 also absorbs light, specifically 700 nanometer red light. This excites its electrons, causing them to jump to a higher energy level and be transferred to another electron acceptor, one with an even higher energy level or more negative redox potential. Step 5, connecting the photosystems. Here's the crucial link. The electrons that came downhill from the PS2 electron transport system don't just disappear, they are passed on to the P700 reaction center in PSI. This replaces the electrons that P700 lost when it got excited. So the electron flow from PS2 essentially fills the hole left in PSI. Step 6, NADPH formation. Finally, the electrons that were excited in PSI and picked up by its acceptor then move downhill again through another short chain. This time, they are ultimately transferred to a molecule of NADP+, nicotinamide adenine dinucleotide phosphate. Along with protons, H+, from the stroma, these electrons are used by the enzyme NADP reductase to reduce NADPH plus H++ NADP. NADPH is a crucial high-energy electron carrier molecule, vital for the later stages of sugar synthesis. So if you visualize this whole pathway, starting from PS2, electrons go uphill in energy, then downhill to the ETS to PSI, then uphill again at PSI, and finally downhill to NADP plus R. That zigzag pattern on an energy diagram gives us the Z shape. This entire process involving both PS2 and PSI is known as non-cyclic photophosphorylation because the electrons flow in one direction from water to NADP plus and are not cycled back. And as we'll see, it generates both ATP and NADPH.
I'm trying to visualize this electron transport. It's quite a dynamic process, lots of moving parts. But if electrons are constantly leaving PSI and moving down that chain, how does PSI keep functioning? It must need to get electrons from somewhere, right? It can't just keep losing them. And where precisely does the oxygen we breathe, the byproduct of photosynthesis, actually come from in all of this?
Those are fantastic, insightful questions, and they lead us directly to one of the most critical aspects of the light reactions, the splitting of water, also known as photolysis. You've hit upon the key point. The electrons lost from photosystem 2 must be continuously replaced for the process to continue. It would stop very quickly otherwise. This essential replenishment is achieved by electrons that become available from the splitting of water molecules. Yes, water. This process of water splitting is directly associated with PS2 and physically takes place on the inner side of the thylakoid membrane, the side facing the thylakoid lumen, the space inside the thylakoid. Let's break down the photolysis reaction. It's quite simple chemically, but profound biologically. A molecule of H2O is split into 2H+, which are protons or hydrogen ions, O, a nascent oxygen atom, which is highly reactive, and 2E, 2 electrons. Sometimes you see it written for 2 water molecules yielding 4H plus O2 and 4E. Now, to address your specific points. 1. The electrons E produced from water splitting are immediately utilized to replace those electrons that PS2 lost when its P680 reaction center was excited. So water is the ultimate electron donor for PS2, making this entire Z scheme possible. 2, the protons, H+, accumulate within the thylakoid lumen. This is very important as they contribute significantly to the proton gradient we'll discuss next, a vital energy store. 3. The nascent oxygen atoms O from two water molecules combine to form molecular O2 gas, O plus O-2. This O2 is then released as a gaseous byproduct of photosynthesis. This right here is the source of the atmospheric oxygen that sustains aerobic life on Earth. So yes, the oxygen you are breathing right now literally comes from the water molecules that plants split using sunlight. Pretty remarkable, isn't it? Okay, so we know electrons flow, water splits, oxygen's released, and NADPH is formed. But what about the other energy currency, ATP? How is that synthesized? Its synthesis in chloroplast is called phosphorylation, and since it's driven by light, we specifically call it photophosphorylation. But how exactly is ATP synthesized? For that, we turn to the elegant and highly efficient chemiosmotic hypothesis. Proposed by Peter Mischel, it explains ATP synthesis not just in chloroplast, but also in mitochondria. This is a frequently tested concept, so let's break it down step by step. The core principle is this. ATP synthesis is linked to the development of a proton gradient across a membrane. In chloroplast, this critical membrane is the thylakoid membrane. Protons, H+, accumulate inside the thylakoid lumen, creating a high concentration there compared to the stroma outside. So what causes this proton gradient? Why do protons build up inside the lumen? There are three main contributing factors. Step 1, proton release from water splitting. As we just discussed, the splitting of water, photolysis, happens on the inner side of the thylakoid membrane. This directly releases protons, H+, into the thylakoid lumen, adding to the concentration inside. Step 2, proton pumping by electron carriers. As electrons move through the electron transport system between psi and psi, certain carrier molecules, like plastoquinone, act as proton pumps. They pick up a proton from the stroma outside as they accept an electron, and then release the proton into the lumen inside as they pass the electron on. This actively transports protons from the stroma into the lumen against their concentration gradient. Step 3. Proton consumption in stroma for NADPH synthesis. Remember the enzyme NADP reductase. It's located on the stroma side of the membrane. To make NADPH plus H plus from NADP plus N, it uses electrons coming from psi and protons from the stroma. By consuming protons from the stroma, this reaction further decreases the proton concentration there. The result, because of these three processes working together, protons released inside from water splitting, protons pumped inside during electron transport, and protons consumed outside during NADPH formation, there's a significant buildup of protons inside the lumen. This creates a steep proton gradient, also called proton motive force, across the thylakoid membrane. The lumen becomes acidic, low pH, and positively charged compared to the stroma. This gradient represents stored potential energy, like water behind a dam. Now, how does this proton gradient lead to ATP synthesis? The stored energy is released when protons flow back down their concentration gradient, from the high concentration in the lumen to the low concentration in the stroma. They can't just diffuse across the membrane, they flow through a special channel. This channel is part of a remarkable protein complex embedded in the membrane called ATP synthase. ATP synthase has two main parts, CF0. This part is embedded within the thylakoid membrane and forms a channel through which protons flow. CF1. This part protrudes out into the stroma. As protons flow through the CF0 channel, it causes the CF1 part to rotate, like a tiny molecular motor. This rotation causes a conformational change in CF1, activating its enzymatic sites. This activation allows CF1 to bind ADP, adenosine diphosphate, and inorganic phosphate, pi, from the stroma and synthesize energy-rich ATP. So, in essence, chemiosmosis requires a membrane, thylakoid, a proton pump, ETS, a proton gradient, and the ATP synthase enzyme. Energy from light is used to build the gradient, and the flow of protons down the gradient through ATP synthase powers ATP production, a truly elegant biological machine. Building on that, it's important to understand that there are two primary modes of photophosphorylation, based on how the electrons flow. First, we have non-cyclic photophosphorylation. This is the main Z-scheme pathway we've been discussing. It occurs when both photosystem I, PSI, and photosystem II, PS2, work in series, connected by that electron transport chain. Electrons flow in one direction, from water through PS2, then PSI, and finally reducing NADP plus to NADPH. This process results in the production of both ADP and NADPH plus H. This is the most common form, providing both energy, ATP, and reducing power, NADTH, needed for the next phase. Then there's cyclic photophosphorylation. This is a bit different. Here, only photosystem I, PSI, is functional. The excited electrons from PSI, instead of going to NADP plus A, are cycled back to the electron transport chain that connects PSI and PSI, and then eventually return to PSI itself. This process typically occurs in the stromal lamellary membranes, which often lack PS2 and the NADP reductase enzyme, the key outcome. Cyclic slow generates only ATP with no NADPH being formed and no water being split, so no oxygen is released either. Why would the plant do this? Well, it's important when the cell needs extra ATP, maybe more than NADPH. For example, the Galvin cycle, which we'll discuss next, requires more ATP than NADPH. Cyclic photophosphorylation helps meet this extra ATP demand. It can also happen when light wavelengths beyond 680 millimeters are predominant, as only PSI can effectively use that light.
Okay, so the light reactions are all about capturing light energy and converting it into chemical energy packets, ATP and NADPH. That's clear now. So how are these energy carriers actually put to use to make the real food, the sugars in the plant? What happens in those dark reactions or carbon reactions?
The light-independent reactions, the Calvin cycle, carbon fixation.
You've articulated the next critical phase perfectly. You're absolutely right. Of the products from the light reactions, ATP, NADPH, and O2 oxygen simply diffuses out of the chloroplast and eventually out of the leaf. Good for us. But ATP and NADPH, those precious high-energy carriers, are immediately channeled into the stroma of the chloroplast, their mission, to power the synthesis of food, or more precisely, sugars. This is what we call the biosynthetic phase of photosynthesis. As we noted earlier, it's also informally termed the dark reaction, but let's reiterate why that's a bit misleading, especially for exams. This phase is not directly driven by light. However, it is entirely dependent on the continuous supply of ATP and NADPH from the light reactions, plus, of course, CO2 and water. Think of it this way. If you suddenly turn off the light, the biosynthetic process would continue for only a very short time, just until the existing ATP and NADPH pool runs out. Then it would stop. When light becomes available again and ATP and NADPH are regenerated, the synthesis restarts. So while it doesn't directly use light photons, it certainly can't happen for long without the products of the light reactions. That dependence is key. Our detailed understanding of this biosynthetic phase largely stems from the brilliant work of Melvin Calvin and his colleagues, Andrew Benson and James Bassam, back in the 1940s and 50s. Using the then new tool of radioactive carbon-14, 14C, as a tracer in experiments with green algae, chlorella, they painstakingly mapped out the entire pathway. This groundbreaking research, which earned Calvin the Nobel Prize, led to the discovery of the very first stable CO2 fixation product. Now let's talk about that primary acceptor of CO2 and the cycle that, quite rightly, bears Calvin's name. Through his meticulous experiments, Melvin Calvin discovered that the first stable product formed after atmospheric CO2 was fixed was a 3-carbon organic acid. Specifically, this was identified as 3-phosphoglyceric acid, 3-PGA. This fundamental discovery is why the pathway is often known as the C3 pathway, because the first stable product has 3 carbons. Now, here's an interesting twist in the story of discovery. Initially, because the first product had 3 carbons, scientists logically assumed that the molecule accepting the CO2 must be a 2-carbon compound, 2C plus 1C and a 3C. They spent years searching for this hypothetical 2-carbon acceptor, but couldn't find it. It was quite a puzzle until further studies unexpectedly revealed that the actual primary CO2 acceptor molecule was, in fact, a 5-carbon keto sugar called ribulose 1-mer5 bisphosphate, RuBP, 5C plus 1C-mer, 6C intermediate middle 2 3C molecules. That was the breakthrough. Melvin Calvin and his team then worked out the entire pathway, demonstrating that it operates in a cyclic manner. And crucially, the CO2 acceptor molecule, RoBP, is continuously regenerated, allowing the cycle to turn again and again, fixing more CO2. This complete pathway is now universally known as the Calvin cycle, or sometimes the Calvin-Benson cycle, or the C3 pathway. And here's a vital point for your exams, a really key concept. The Calvin pathway operates in all photosynthetic plants, every single one. It doesn't matter if a plant is classified as C3 or C4, which we'll get to. The Calvin cycle is the fundamental universal pathway for the actual synthesis of sugars from CO2. For ease of understanding and strong exam recall, we can break down the Calvin cycle into 3 main stages. Think of it as 3 phases: carboxylation, reduction, and regeneration. Stage 1, carboxylation. This is arguably the most critical initial step. Carboxylation simply means adding a carboxyl group, COH, which comes from CO2. It's the fixation of atmospheric CO2 into a stable organic intermediate. In this reaction, 1 molecule of CO2 combines with 1 molecule of the 5-carbon acceptor, ribulose 1-val5 bisphosphate, RuBP. The enzyme that catalyzes this crucial reaction is incredibly important. It's called RuBP carboxylase oxygenase, or more commonly, Rubisco. It's essential to highlight that Rubisco is not just vital, it is in fact believed to be the most abundant protein on Earth. A common exam factoid. This carboxylation reaction results in an unstable 6-carbon intermediate that immediately splits into 2 molecules of 3-phosphoglyceric acid, 3-PGA, our 3-carbon product. Stage 2, reduction. This stage involves a series of reactions where the 3-PGA is converted into sugars. It's called reduction because electrons and protons are added, primarily from the NADPH generated during the light reactions. This is the main energy-consuming part of the cycle. For every single molecule of CO2 fixed, this reduction phase uses 2 molecules of ATP for phosphorylation, activating the 3-PGA, and 2 molecules of NADPH for the actual reduction step, forming a 3-carbon sugar phosphate, glyceraldehyde 3-phosphate, or G3P. To synthesize just 1 molecule of glucose, a 6-carbon sugar, which is often considered the end product, the Calvin cycle needs to complete 6 turns, fixing 6 molecules of CO2. Why 6? Because glucose has 6 carbons and each turn fixes 1 carbon. Stage 3, regeneration. This final stage is absolutely critical. For the cycle to continue, the starting molecule, the CO2 acceptor, ρBP, must be regenerated from the G3P molecules produced in the reduction phase. Some of the G3P goes towards making glucose or other sugars, but most of it is used to reform ρBP through a complex series of reactions. This regeneration process also requires energy. It consumes an additional 1 molecule of ATP for phosphorylation for every CO2 molecule that was fixed. So let's tally up the energy cost for the Calvin cycle, a very common exam question. For every 1 CO2 molecule fixed, carboxylation uses 0 ATP, 0 NADPH. Reduction uses 2 ATP, 2 NADPH. Regeneration uses 1 ATP, 0 NADPH. Total per CO2, 3 molecules of ATP and 2 molecules of NADPH. And therefore, to synthesize 1 molecule of glucose, which requires fixing 6 CO2 molecules, total for 1 glucose, 18 molecules of ATP is 6 turns by 3 ATP, and 12 molecules of NADPH, 6 turns by 2 NADPH turn. Notice the ratio, 3 ATP to 2 NADPH per CO2 fixed. This higher ATP requirement is one reason why cyclic photophosphorylation, which only makes ATP, is important. It helps provide that extra ATP needed to keep the cycle balanced.
That's a remarkably clear breakdown of the Calvin cycle: carboxylation, reduction, regeneration, and the energy costs.
Okay, so it's the universal pathway for making sugars. But you also mentioned C4 plants exist. If they still rely on the Calvin cycle in the end, how exactly are they different? What makes them special? What's the C4 pathway about?
Adapting to conditions of the C4 pathway and photorespiration.
You've hit on exactly the right next point. Because while the Calvin cycle is universal, the way some plants initially capture CO2 and deliver to that cycle is brilliantly different, especially as an adaptation to challenging environments. The C4 pathway is a remarkable evolutionary strategy, primarily seen in plants adapted to hot, dry, sunny conditions. Think dry, tropical regions. Common examples include maize, corn, sugarcane, sorghum, and many tropical grasses. You're spot on that while their initial CO2 fixation product is a 4-carbon acid, typically oxaloacetic acid, or OAA, they do in fact still utilize the C3 pathway, the Calvin cycle, as their main biosynthetic route for synthesizing sugars. So the Calvin cycle still happens, but somewhere specific. So what sets them apart? Why evolve this extra pathway? C4 plants are truly special and have several key distinguishing characteristics that give them significant advantages, particularly in those hot, dry climates. 1, they possess a unique leaf anatomy known as Krantz anatomy. We'll look at that. 2, they exhibit a much higher tolerance to elevated temperatures. They can keep photosynthesizing efficiently when C3 plants might struggle. 3, they show a more robust photosynthetic response to high light intensities. They don't saturate as easily. 4, crucially, they largely avoid photorespiration, a process we'll discuss that can be quite wasteful for C3 plants under those same hot, dry conditions. 5, as a direct result of these adaptations, they often demonstrate significantly greater productivity and biomass yield compared to C3 plants, especially in warm climates. That's why crops like sugarcane and maize are so productive. Let's first talk about this unique leaf anatomy, Krantz anatomy. The term Krantz is German for wreath, and it perfectly describes the arrangement of cells. If you were to take a cross-section of a C4 plant leaf, like maize, and look under a microscope, you'd see a distinctive feature, particularly large bundle sheath cells. These cells form one or more dense layers tightly packed around the vascular bundles, the veins of the leaf, creating this wreath-like pattern. These bundle sheath cells aren't just large, they have specific features vital to the C4 mechanism. They contain a large number of chloroplasts, often larger than those in mesophyll cells. Their cell walls are unusually thick and impregnated with suberin, making them relatively impervious to gaseous exchange, especially O2 escaping or CO2 leaking out. They sort of seal the compartment. Significantly, they often have few or no intercellular air spaces between them, ensuring close contact. It's this specialized Krantz anatomy that strategically sets the stage for the C4 pathway, creating 2 distinct types of photosynthetic cells working together: the outer mesophyll cells and the inner bundle sheath cells. Okay, now let's trace the steps of this unique preliminary carbon fixation pathway, known as the Hatch and Slack pathway, named after the scientists who discovered it. It's another cyclic process, but it happens before the Calvin cycle. acting as a clever CO2 concentrating mechanism. This is critical for your exams. Step 1. Primary CO2 acceptance in mesophyll cells. The journey begins in the cytoplasm of the mesophyll cells, the outer layer of photosynthetic cells in C4 leaf. Here, the primary CO2 acceptor is not rho BP. Instead, it's a 3-carbon molecule called phosphenol pyruvate, PP. Atmospheric CO2 enters the leaf and dissolves to form bicarbonate, HCO3, which then combines with PP. This reaction is catalyzed by a highly efficient enzyme called PP carboxylase, or pepcase. It's absolutely critical to note. Pepcase has a very high affinity for bicarbonate, and importantly, it does not bind with oxygen. Also, the mesophyll cells of C4 plants generally lack the Rubisco enzyme. That's the key difference from C3 plants. When bicarbonate combines with PP, it forms a 4-carbon organic acid, oxaloacetic acid, OAA. This is the first stable product, hence the name C4 pathway. Step 2. Conversion and transport. The OAA formed in the mesophyll cells is unstable and quickly converted into other, more stable 4-carbon compounds, typically malic acid or aspartic acid, still within the mesophyll cells. These 4-carbon acids are then actively transported from the mesophyll cells into the adjacent bundle sheath cells, moving through plasmodesmata, those small channels connecting plant cells. Step 3. Decarboxylation in bundle sheath cells. Once inside the cytoplasm or chloroplast of the bundle sheath cells, these 4-carbon acids undergo decarboxylation. This means they are broken down enzymatically to release CO2 and regenerate a 3-carbon molecule, usually pyruvate. This is the core of the C4 strategy. CO2 is released at a very high concentration right inside the bundle sheath cells, baiting the Rubisco enzyme located there. Remember, bundle sheath cells are rich in Rubisco but lack pepcase. Step 4. Calvin cycle and regeneration. The CO2 that's just been released at this high concentration in the bundle sheath cells immediately enters the C3 Calvin pathway, which operates within the chloroplast of these bundle sheath cells. This is where the actual synthesis of sugars like glucose takes place, using the CO2, along with the ATP and NADPH generated from the light reactions, which also occur in bundle sheath chloroplast, so sometimes slightly modified. Meanwhile, the 3-carbon molecule pyruvate that was left after decarboxylation is transported back to the mesophyll cells. In mesophyll cells, this pyruvate is converted back to PEP using ATP in the process. This regenerates the initial CO2 acceptor, completing the C4 cycle and allowing it to continue. So the key takeaway: C4 plants use a clever spatial separation of steps. Initial CO2 capture by pepcase happens in the mesophyll cells. The 4-carbon acid then travels to the bundle sheath cells, where it releases CO2 at a high concentration, allowing Rubisco to efficiently run the Calvin cycle there without being bothered much by oxygen, because it's a CO2 pump, essentially.
That's a truly brilliant strategy. It really does sound like they found a way to pump CO2 to where it's needed most. You mentioned that C4 plants largely avoid photorespiration. Now I think I see why. But could you explain exactly what is photorespiration and why is it considered a problem or wasteful for C3 plants? Is it always bad?
You've connected the dots perfectly. That CO2 concentrating mechanism is exactly why C4 plants minimize photorespiration. Now, let's unpack photorespiration itself, as it's a crucial concept, especially for comparing C3 and C4 efficiency. Photorespiration happens because of that dual nature of Rubisco we mentioned. Remember, Rubisco stands for ribulose 1,5-bisphosphate carboxylase oxygenase. It can act as both a carboxylase, adding CO2 to RuBP, and an oxygenase, adding O2 to RuBP. The binding is competitive. It depends on the relative concentrations of CO2 and O2 at the enzyme's active site, and also on temperature. Higher temperatures tend to favor the oxygenase reaction. Now let's look at what happens in C3 plants under certain conditions, typically highlight high temperature and low internal CO2 concentration. These conditions often arise when stomata close to conserve water. Under these conditions, the O2 concentration inside the leaf builds up from photosynthesis, while the CO2 concentration drops as it's being used up and entry is restricted. This shift of the CO2-O2 ratio means O2 starts to bind more frequently to Rubisco's active site. When O2 binds, Rubisco acts as an oxygenase. Instead of fixing CO2 with RoBP to form 2 molecules of 3-PGA, the start of the productive Calvin cycle, RoBP combines with O2 to form 1 molecule of 3-phosphoglycerate, 3-carbon, and crucially 1 molecule of phosphoglycolate, a 2-carbon compound. This 2-carbon phosphoglycolate cannot directly enter the Calvin cycle. It has to be salvaged or metabolized through a complex pathway called photorespiration. This pathway involves reactions in the chloroplast, peroxisome, and mitochondria. Here's why photorespiration is considered wasteful or detrimental to the net efficiency of C3 plants. It results in no net gain of fixed carbon. In fact, during the photorespiratory pathway, some of the carbon that was initially fixed is released back as CO2 in the mitochondria. So it literally undoes some of the work of photosynthesis. It consumes energy. The pathway requires ATP in reducing power like NADH, indirectly related to NADPH, effectively using up valuable energy products generated by the light reactions without producing any sugar in return. It leads to a net loss of carbon and energy from the photosynthetic process, reducing the overall efficiency, sometimes by as much as 25% or more, in C3 plants under hot, dry conditions. So is it always bad? Well, its primary consequence is reduced photosynthetic yield. However, some scientists suggest it might play minor protective roles, like dissipating excess energy under high light or contributing to certain metabolic pathways. But from an agricultural perspective, focusing on yield, it's generally seen as a major limitation for C3 crops in warm climates. And as you correctly deduced, C4 plants avoid this problem. Their C4 pathway acts as that CO2 concentrating mechanism. By pumping CO2 into the bundle sheath cells and releasing it there, they maintain a very high CO2 concentration right around the rubisco enzymes. This high CO2 level ensures that rubisco overwhelmingly functions as a carboxylase, binding CO2, and rarely acts as an oxygenase. The oxygenase reaction is effectively suppressed. That's the fundamental reason why C4 plants thrive in hot, bright conditions, where C3 plants suffer from significant photorespiratory losses. It's a key adaptation for productivity in those environments. Given how important the C3 versus C4 comparison is for your exams, let's quickly summarize the key differences again in a table-like format, hitting the main points. Feature C3 plants, C4 plants, Calvin cycle, location mesophyll cells. Bundle sheath cells, initial CO2 fixation, mesophyll cells fortis. Primary CO2 acceptor, RoBP, 5C-PGP, 3C. Primary CO2 fixation product, PGA, 3C, OAA, 4C. Enzyme for initial fixation, rubisco PP carboxylase. Location of rubisco mesophyll cells. Location of pukate absent mesophyll cells. Lanson-Krantz anatomy. Absent present high cell. Photorespiration high present. Negligible absent high. CO2 saturation point higher. 450 1 lower. Temperature optimum lower. 20-25 degrees C. Higher. 30-20 degrees C. Fire. 30-40 degrees C. Examples: wheat, rice, potato, soybean, maize, sugarcane, sorghum. Keep these distinctions clear in your mind. They are very frequently tested.
Factors affecting photosynthesis, optimizing the process.
Okay, so we've thoroughly explored the incredible mechanisms of photosynthesis, both C3 and C4. Now, it's essential to understand that the rate at which photosynthesis occurs isn't constant. It can vary quite a bit. And therefore, the plant's growth and ultimate yield, especially important for crops, isn't static either. It's influenced by several factors. We can broadly categorize these factors into two groups: internal or plant factors and external or environmental factors. Internal factors are those inherent to the plant itself. These include things like the number, size, age, and orientation of its leaves. Are they positioned to catch light well? The quantity and efficiency of mesophyll cells and the chloroplasts within them, the internal concentration of CO2 within the leaf spaces, and of course the total amount of chlorophyll and other pigments present. These factors are largely determined by the plant's genetic makeup and its specific stage of growth and development. External factors, on the other hand, are conditions in the plant's environment. These include the availability of sunlight, its intensity, quality, wavelength, and duration, the ambient temperature, the CO2 concentration in the atmosphere, and the availability of water. It's important to remember that as a plant photosynthesizes, all these factors are simultaneously at play. However, there's a crucial principle that governs how they interact to determine the overall rate. This is Blackman's law of limiting factors, proposed way back in 1905. The law states, essentially, if a chemical process is affected by more than one factor, then its rate will be determined by the factor which is nearest to its minimal value. It is the factor which directly affects the process if its quantity is changed. End quote. What does that mean in simpler terms? It means that at any given moment, the rate of photosynthesis is limited by the slowest factor, the one that's in shortest supply relative to the plant's needs. Even if all other factors are optimal, the rate won't increase unless that specific limiting factor is increased. Let's use a simple analogy. Imagine you're assembling cars on a production line. You need chassis, engines, and wheels. Even if you have hundreds of chassis and engines ready, if you only have enough wheels for 10 cars per hour, you can only make 10 cars per hour. The wheels are the limiting factor. Similarly, in photosynthesis, if a plant has plenty of water and sunlight, but the CO2 concentration is very low, the rate will be limited by CO2. Increasing light or water won't help much until CO2 levels rise. Let's look at some of these specific external factors in more detail, as their effects are often tested. 1. Light. When we discuss light, we consider its quality, wavelengths, intensity, brightness, and duration. Let's focus on intensity. At low light intensities, there's typically a linear relationship between the amount of light shining on the leaf and the rate of CO2 fixation. More light means faster photosynthesis up to a point. However, as light intensity continues to increase, the rate gradually begins to slow down and eventually saturates or plateaus. At this point, light is no longer the limiting factor. Something else, like CO2 availability or the capacity of the enzymes, is holding the rate back. Here's an interesting fact. Light saturation for many plants actually occurs at only about 10% of full direct sunlight. This might surprise you. It means that for many plants out in the open, light intensity itself is rarely the primary limiting factor during the brightest parts of the day. Exceptions might be plants growing in deep shade or under a dense forest canopy. One important caution, though. Excessively high light intensity can actually damage the photosynthetic machinery, causing photoinhibition and breakdown of chlorophyll, which decreases the rate. So too much light can be bad too. 2. Carbon dioxide concentration. This is a major one, especially in the context of agriculture and climate change. The concentration of CO2 in the atmosphere is naturally very low, currently around 0.04%, or 420 parts per million. For many plants, particularly C3 plants, this low atmospheric concentration makes CO2 often the major limiting factor for photosynthesis under normal daylight conditions. An increase in CO2 concentration, say up to about 0.05 or 0.1%, can significantly boost CO2 fixation rates in plant growth, especially for C3 plants. However, very high concentrations, sustained over long periods, can sometimes have negative effects. Now remember our C3 and C4 plants. They respond differently to increasing CO2. Under low light, neither group responds much light is limiting, but at high light intensities. C4 plants show an increase in rate, but they tend to reach CO2 saturation at concentrations not far above current atmospheric levels, around 300 and 6,400. Their CO2 pump is already efficient. C3 plants, however, continue to show increased photosynthetic rates as CO2 levels rise, often saturating only at much higher concentrations, beyond 450 SORL1, sometimes much higher. This clearly indicates that current atmospheric CO2 levels are often limiting for C3 plants' potential productivity, but less so for C4 plants. This knowledge has practical applications. It's why commercial greenhouses growing C3 crops like tomatoes, cucumbers, or bell peppers often use CO2 enrichment, adding extra CO2 to the air, to boost yields significantly. 3. Temperature. The dark reactions, the Calvin cycle, are driven by enzymes, and enzyme activity is highly sensitive to temperature. Therefore, the rate of the dark reactions, and thus overall photosynthesis, is significantly temperature controlled. Light reactions are also somewhat temperature sensitive, but generally less so than the enzymatic dark reactions. Each plant species has an optimum temperature range for photosynthesis. Below this range, rates are slow. Above it, enzymes can start to denature, and rates drop sharply. Again, C4 and C3 plants differ. C4 plants generally have a higher temperature optimum, often performing best around 30–40 degrees C, making them well suited to tropical climates. C3 plants typically have a lower optimum, often around 20–25 degrees C, thriving in more temperate regions. The specific optimum always depends on the plant's adaptation to its native habitat. 4. Water. Water is, of course, a direct reactant in the light reactions, remember photolysis. However, its effect as a limiting factor on the rate of photosynthesis is often more indirect. When a plant experiences water stress, lack of sufficient water, it triggers several physiological responses that negatively impact photosynthesis. The most immediate effect is stomata closure. To conserve water, the plant closes the tiny pores, stomata, on its leaves. But these pores are also the entry point for CO2. So closing stomata severely reduces the availability of CO2 inside the leaf, directly limiting carbon fixation. This is often the primary way water stress limits photosynthesis. Secondly, severe or prolonged water deficit can cause leaves to wilt. Wilting reduces the effective surface area of the leaves exposed to sunlight, decreasing light capture. It also generally slows down metabolic activity within the leaf cells. So, while water is essential as a reactant, its scarcity often limits photosynthesis mainly by restricting CO2 supply and affecting overall plant health and function. Outro. So we completed our deep dive into the incredible, absolutely essential process of photosynthesis in higher plants. Quite a journey, wasn't it? We started with the basics. Photosynthesis as a physicochemical process making plants autotrophs, the ultimate source of our food and atmospheric oxygen. We traced the fascinating history from Priestley's bell jars and Ingenhousz's bubbles to Sachs locating chloroplasts, Engelmann mapping the action spectrum, and Van Niel's crucial insight about oxygen coming from water. Then we dissected the machinery, the chloroplast structure, the thylakoids and stroma, and the vital division of labor. We meticulously detailed the light-dependent reactions, pigment absorption, the PSII and PSI partnership in the Z scheme, photolysis releasing O2, and the brilliant chemiosmotic mechanism generating ATP and NADPH. We also clarified non-cyclic versus cyclic photophosphorylation. Following that, we moved to the light-independent reactions in the stroma, how ATP and NADPH power the universal Calvin cycle, involving carboxylation by rubisco, reduction to form sugars, and regeneration of rub-EP. We explored the clever adaptations of C4 plants, their unique Krantz anatomy, and the Hatch-Slack pathway acting as a CO2 pump to overcome photorespiration, giving them an edge in hot, dry climates. And finally, we discussed how the rate of this whole process is governed by limiting factors like light, CO2, temperature, and water, always following Blackman's law. Now, this chapter, Photosynthesis in Higher Plants, truly is a cornerstone of plant physiology. It's absolutely vital for your class 11th and 12th board exams, and definitely a high-weightage topic for NEET. It beautifully combines fundamental concepts with intricate molecular mechanisms. My strong advice to you is to really revise these pathways, especially the Z-scheme, the steps of chemiosmosis, and the stages of the Calvin cycle. Don't just read them. Try actively drawing them out, step by step, maybe on a whiteboard or just rough paper without looking at your notes at first. Test yourself. This active recall is incredibly powerful for locking it into memory. Also, make sure you practice plenty of past year questions. Look specifically for questions comparing C3 and C4 plants, questions about rubisco's dual role in photorespiration, and scenarios involving limiting factors. These are classic, frequently tested concepts. Remember, truly understanding the why and how behind these processes, not just memorizing facts, will not only help you score better, but also give you a much deeper appreciation for the sheer elegance and complexity of life itself. If any doubts still linger after you revise, please don't hesitate to reach out. Keep up your focused efforts, keep learning, and keep growing.
Thank you so much, tutor. That was incredibly clear, detailed, and really well-structured. I feel like I've truly understood the intricate dance of photosynthesis now, not just, memorize the steps. I definitely feel much more confident tackling questions on this for my exams.